Hello and welcome back to our continuing journey of chapter 3. We're going to talk about membranes. Now, hopefully a lot of the topics in here is going to be review, but I just wanted to throw this little mini lesson out there if it's been a while since you've had your general biology, but it's always good to review if you just came out of Bi 211 as well. All right, so plasma membranes. If you remember, they are phospholipid bilayers, and just to review what a phospholipid is, right? They've zoomed one out right here. We had a hydrophobic head, which was polar, right? So it had some positive and negative charges. And we had a hydrophilic tail, which was neutral, it was non-polar. It didn't have any charges. So when these molecules, they kind of self-arrange with the hydrophilic heads pointing towards the watery environments, either outside of the cell or inside of the cell, and then those hydrophobic tails kind of pointing towards each other, right? So this is our neutral environment, and this is our polar environment. Whoops. Let me get my pluses and minuses right. So we have a watery environment out here and a watery environment out here. All right, so hopefully that's ringing a bell. And then because of that selective permeability, the cell needs to be able to get things in and out of it. So we typically are going to use integral proteins that are channels here. We also, because the plasma membrane is part of the outer layer, a lot of times we have some polysaccharides or glycoproteins or glycolipids. Glyco is just sugar, right? So a sugar combo protein, a sugar combo lipid. These can be used for signaling or communication. If you've heard of antigens before, the glycoproteins could be considered antigens, which are like the recognizing part of a pathogen. And then remember, peripheral proteins are just proteins that are found on the surface of the phospholipid bilayer, not passing all the way through the hydrophobic middle part. Okay? And then integral proteins can have some hydrophobic components, allowing them to be integrated into that lipidy part of the phospholipid bilayer. So hopefully that's a good review for you. It's kind of coming back. Oh yeah, phospholipid bilayer, selectively permeable, made up of these amphipathic phospholipids, allowing things to move through freely or through an ion channel or a transport channel. Okay. So you should also be somewhat familiar with what we call an electrochemical gradient. Because of that selective permeability of the plasma membrane, it can help to create differences in concentrations of things and differences in charges of things. So in this diagram, we're showing primarily just sodium and chlorine, which are two very common ions that you might find inside and outside of cells. So we have the orange little circles are sodium. And if we look, we have a whole bunch of sodium on the outside of the cell and very little sodium on the inside of the cell. So it's high out here and it's low inside. So this is what we call a chemical gradient. So we have a separation of concentration of things. And then it's the reverse for our chlorine. So we have a whole bunch of chlorine inside of the cell and just a few chlorine ions outside of the cell. So here our chlorine is low on the outside, but high on the inside. And because of that difference, that is another chemical gradient. So those two ions are representing the chemical part of the electrochemical gradient. Then if we take a look at the summation of the electrical charges, proteins and DNA tend to be negatively charged. And there's a lot of those inside of cells. Plus we had a good collection of the negatively charged chlorine ions. So what that typically means is the inside of this phospholipid bilayer is going to be overall negative in charge. Comparing that to the outside, it's not so much saying that the outside is positive, it's just saying that the inside is more negative, and it typically hovers around a negative 70 millivolts. If you've taken A&P, you know exactly what I'm talking about. This is the charge that we saw in the conduction of action potentials. We started off as a resting state of negative 70 millivolts, and then as that changed, when we got signaling coming in from acetylcholine or somewhere else, we changed that electrochemical gradient, this transmembrane potential, and that helped us trigger the cell communication. So all we're doing here is just reminding you that the inside of the cell is a little bit more negative than the outside of the cell. Most likely, or not most likely, they know, it is because of the negative charges on the DNA and proteins, in addition to any negatively charged ions. There's just more of those inside of the cell. So this is what we call our electrochemical gradient, just another characteristic that's associated with plasma membranes. So then we can talk and review a little bit about transport processes. There's two major categories. We have what's called passive and active. So if you remember, passive processes do not require the cell to exert any energy to move things across the plasma membrane. Active transport, you need energy to move things across the plasma membrane. And in passive processes, we are always going from a high concentration gradient to a low concentration gradient. And it could be that electrochemical gradient. The concentration gradient, part of that is going from high to low. So we have a couple different examples of this passive transport. In the first one over here in picture A, this is diffusion. Simple diffusion as substances moving directly through the phospholipid bilayer, and for them to do that, they have to be nonpolar. So remember, nonpolar is also the same thing as hydrophobic or uncharged or kind of a neutral charge. So only nonpolar things can simply diffuse through that phospholipid bilayer. Anything else, so if it's large, if it's got a charge on it, so all of these things are polar. They have to be polar if they're going to be going through these, what we call facilitated diffusion. So we have facilitated diffusion through a non-specific channel, so it really doesn't care as long as the size fits, it's fine. Facilitated diffusion through a specific channel is only good for that one thing. In this case, it looks like it's glucose or some kind of carbohydrate, but we have some facilitated diffusion channels that only allow sodium to come through or chlorine to come through or bicarb or ammonia or whatever the ion is. It's specific to just that one thing. And a special type of diffusion or passive transport is what we call osmosis. So osmosis is the diffusion of water. So water moving from a high concentration to a low concentration to reach equilibrium. So here's our osmosis term. And osmosis, water can move in a couple different ways. We have through a channel, and these are called aquaporins. You may remember that from other classes. So aquaporins is a channel specific for water. And that's where most of the water moves through a plasma membrane because water is polar, right? It's got the Mickey Mouse kind of look. It's negative on the oxygen side. It's positive on the hydrogen side, right? It's a polar molecule. So it mainly travels through those aquaporins, but a few molecules can diffuse. They're not very readily because they're polar and the inside of the plasma membrane is hydrophobic. But we have to appreciate that there's some water molecules that can move simply through diffusing through the plasma membrane, but also note the majority of water molecules moving through a plasma membrane are going to be moving through aquaporins. So all of these processes, simple diffusion, facilitated diffusion, and osmosis are all passive. We do not need energy because things are going from their high concentration to low. It's entropy, it's all kind of following energy going from exergonic reactions, right? Releasing energy in that process. Along with the idea of osmosis, we want to make sure you guys also remember your terms isotonic, hypertonic, and hypotonic. So remember, isotonic is equal. You have the equal concentrations of solutes inside and outside of the cell. Hypertonic, right? The root hyper means more. So in hypertonic solutions, you have more solute in the solution, and hypotonic, hypo meaning below or less solute concentration. So in an isotonic solution, water will be osmosing equally in both directions, and the cell's not going to be negatively impacted by this. If you place a cell in a hypertonic solution, because there's more solute outside, water follows salt. So water wants to go towards the higher solute concentration. So if you place a cell, like a freshwater cell, into a really salty situation, water's going to want to leave, and so the cell's going to shrink. If you place that same kind of cell in a hypotonic solution, there's more water outside of the cell, which means the inside of the cell is hyper. It's going to have more solids. And so water is going to enter inside of the cell and it will keep rushing into the cell until the cell swells and either bursts if it's like an animal cell with no protective cell wall, or if you do have a cell wall like a plant or a fungus or bacteria, it swells up and pushes and the cell wall helps to protect from bursting. But it still could also negatively impact the cell because it's diluting its concentration of electrolytes inside. All right, so this should be review from your general bio. If you have questions over this topic, because I know it can be kind of flippy floppy, like which one's which, send me an email, stop by an office hour, and I'll help try to straighten it out for you. Okay, so those are just a specific touch base with osmosis and water moving towards higher solute concentration. And then lastly, we have active transport. So remember, active transport is going from a low concentration to a high. And to do that, we're going against that normal flow of directionality. So we need to burn energy to do that. And it's usually in the process of breaking apart an ATP. So there's a couple different forms, just like we had different forms of passive transport. We have what's called ATPase uniport, which is a kind of a big title just to say you're using ATP to pump one thing against its concentration gradient. Antiport, sometimes also called counter transport. This is what you know best as the sodium-potassium pump. You may be familiar with that. So remember, sodium gets pumped out, potassium gets pumped in to maintain that electrochemical gradient. So we're burning the molecule of ATP to pump the black circle against its concentration gradient and the green square against its concentration gradient. So this is antiport or counter transport. In the next example, this symport or coupled active transport it's a little bit of a combination of active and passive. We are burning energy, pumping the black circle against its concentration gradient, but then we have another channel in close proximity that's going to take advantage of this new high to low concentration gradient, and with it, it's going to bring something else against its concentration gradient. This second channel is free in that we're not spending energy to move the green square against its concentration gradient because it's kind of coupled with the black circle going down its concentration gradient. But if we didn't burn ATP here to make that black circle concentration gradient to begin with, we wouldn't be able to do the coupled reaction. Okay. And then lastly, we have what's called group translocation, which is found primarily in bacteria. So the idea behind this is when we take a look at counting concentrations, we're counting the same thing, right? So if you have glucose and you have a whole bunch of glucose inside of the cell, that would be moving glucose against its concentration gradient. And it would be hard to do that. You'd have to burn energy to do that. But in group translocation, they kind of trick the cell because what they do is they add a phosphate group right here to that sugar. So right, CH2OH is the normal part that's hanging off of that glucose. But then we add this little PO4, the PO4, sorry. So we add that phosphate group, and glucose 6-phosphate is not the same thing as just regular old glucose. So the cell's like, I'll keep on bringing in that glucose because we're not going against our glucose concentration gradient. We're blinded to the glucose 6-phosphate, but it's still considered active transport because where do you think the phosphate group comes from? It comes from ATP. So we take an ATP molecule, break it down, phosphorylate the glucose inside of the cell, which makes the cell still be able to bring in glucose passively, but without burning that ATP, we would not be able to make the glucose 6-phosphates. So it's like an indirect active transport called group translocation. All right, so again, all of this membrane stuff should be relatively a good review for you. But again, if you're feeling a little bit rusty, you can go back and review some of this stuff. If you still have your biology or A&P book, you might be able to review that, watch some YouTube videos, come ask some questions, stop by office hours, send me emails. But you should be comfortable with these concepts because it's going to help us understand some applications in different contexts later on, specifically dealing with bacteria. All right, so that wraps up our plasma membrane lecture. I will see you next time.